Hi there, and welcome to Q English. In this series, we explore the lives of amazing people and help you improve your English at the same time. Ready? Let's dive in. Seeds of Discovery. The Life and Legacy of Barbara McClintock. Chapter 1. Roots in Hartford. Barbara McClintock was born on June 16, 1902, in Hartford, Connecticut, a city known for its libraries and learning. From the beginning, Barbara stood a partner in loud ways, but in the quiet persistence that would shape her future. Her birth name was Eleanor McClintock, but her family soon began calling her Barbara, a name she carried with pride and independence. Her parents, Thomas and Sarah McClintock, were both supportive in different ways. Thomas, a doctor by profession, believed in the power of science and reason. He often shared stories of his work and asked thoughtful questions, which young Barbara would listen to with great interest. Sarah, on the other hand, had artistic talents and a strong sense of discipline. She valued education, but also worried about how Barbara's independent nature would be accepted by society. Barbara was the third of 4 children. Growing up, she often felt like the quiet one in the family. While her siblings played together or followed more traditional paths, Barbara found joy in solitude. She liked observing things closely: flowers, insects, even the way shadows moved across the walls in the late afternoon. These quiet observations planted the first seeds of curiosity that would later bloom into scientific brilliance. When Barbara was still a child, the family moved to Brooklyn. New York life in the city was a big change, but Barbara remained inwardly focused. She didn't need much to be happy, just a notebook, a quiet corner, and something to think about. At school, her teachers quickly noticed her intelligence. Though not talkative in class, she had a deep understanding of every subject she studied. Mathematics and science became her favorites. Her mother, however, was not immediately supportive of Barbara's growing interest in science. At the time, society had strong ideas about what girls should do. Becoming a scientist was not considered ladylike. Sarah worried that Barbara might never marry or fit in if she followed such an unusual path. But Thomas, her father, quietly encouraged her. He gave her books, asked her questions, and most importantly, he believed in her. Despite her mother's concerns, Barbara stayed true to herself. She didn't rebel loudly, but she didn't give in either. Instead, she found ways to pursue her interests within the limits she was given. At Erasmus Hall High School in Brooklyn, she focused on academics. She joined clubs that explored science and nature, and she read constantly, often choosing books far beyond her grade level. One particular memory from childhood stayed with her for years. She once spent an entire afternoon watching how ants built their tunnels in a dirt-filled glass container. While others may have seen it as just a child's play, for Barbara, it was a window into a world of order, patterns, and hidden intelligence. The way the ants worked together, how they adapted, how the same group could rebuild a destroyed tunnel overnight, it all fascinated her. Even then, she was learning that life is full of systems and surprises, just waiting to be discovered. By the time she graduated from high school, Barbara already knew that she wanted to study science more seriously. She was quiet, yes, but also deeply determined. Her path was not going to be easy, especially as a young woman in the early 20th century. But she had already decided she would not be stopped by fear or by other people's expectations. The same deep focus that helped her understand ants, shadows, and numbers would soon guide her into a world that very few had explored before. Her journey to become one of the most important geneticists in history had already begun. Chapter 2. A Curious Mind Blossoms. Barbara McClintock arrived at Cornell University in 1919, full of quiet determination and an eagerness to learn. Ithaca, New York, with its rolling hills and crisp air, became the setting for one of the most important chapters in her life. She enrolled in the College of Agriculture, a choice that might seem surprising at first, but Barbara saw plants, especially maize, or corn, not as ordinary crops, but as puzzles full of mystery and beauty. Corn, with its many colors and patterns, had more to say than most people realized. Barbara would later dedicate much of her life to understanding these silent messengers of genetic information. But in those early days at Cornell, she was just beginning to discover where her curiosity could take her. At first, Barbara studied a little bit of everything: botany, chemistry, zoology. Each subject opened new doors, but it was a course in genetics that truly lit a fire in her mind. The class was taught by Professor Rollins Emerson, a gentleman with a love for maize genetics. His lectures didn't just explain science, they invited students to wonder, to question, and to explore. Barbara found herself completely absorbed. She spent long hours in the university library, reading everything she could find on chromosomes and inheritance. Unlike many of her classmates, she wasn't satisfied with simply passing exams. She wanted to understand how life worked at its deepest level, how traits were passed from one generation to the next, how tiny changes in cells could create such incredible diversity. One evening, while other students relaxed or went into town, Barbara sat at her microscope, studying cornroot tips. The laboratory was quiet except for the soft hum of a lamp and the occasional rustle of notebook pages. Through the lens, she saw chromosomes lining up, dividing, moving. It was like watching a secret dance inside the cells, a dance that told the story of life itself. Her professors noticed her dedication. One of them, Dr. Lester Sharp, became a mentor. He encouraged her to continue her research and invited her to join a small group of graduate students who were working in the field of cytogenetics, the study of chromosomes and their role in heredity. At that time, this was still a young science, and women were rarely invited to participate in serious academic research. But Barbara didn't think of herself as someone who needed permission. She simply worked harder than anyone else. Outside the lab, Barbara remained somewhat of a mystery. She was not interested in fashion or parties. She didn't join sororities or attend dances. Instead, she rode her bicycle across campus, her notebooks often stuffed in her basket. She loved music and occasionally played the banjo, but only when alone. She didn't mind being different. In fact, she seemed to prefer it. Her independence, which had made her a quiet outlier in childhood, now became her greatest strength. As Barbara advanced in her studies, she began doing her own research. She worked with corn plants in the greenhouse and in the fields, analyzing their traits and linking them to the behavior of chromosomes. Slowly, she was building a new understanding of genetics, one that went beyond what anyone had imagined. Still, she faced challenges. Although she earned her bachelor's degree in 1923 and then her master's in 1925, the path to a PhD was not smooth. Many faculty members believed women did not belong in advanced science programs. Some even said so directly. Barbara heard their comments, but she didn't argue. She simply did her work quietly, precisely, and with unshakable focus. In 1927, she received her PhD in botany, specializing in cytogenetics. It was a rare achievement for a woman at the time, but more importantly, it marked the beginning of a lifetime of groundbreaking research. One summer day, as Barbara stood among rows of corn taller than herself, a breeze lifted the tassels and filled the air with a warm, earthy scent. She touched a leaf gently and smiled. This world of living color and quiet complexity was her true classroom. The plants were not just study subjects. They were partners in discovery. Corn, she had learned, could whisper secrets, and Barbara McClintock was ready to listen. Chapter 3. Cornell and the Call of Genetics. Barbara McClintock's time at Cornell did not end with her PhD. In fact, it was only the beginning. While many of her classmates moved on to jobs or other degrees, Barbara stayed. The laboratory, the maize fields, and the sharp-eyed discussions with fellow scientists became the center of her life. Cornell was not just a university, it was a home for her growing scientific vision. In the late 1920s, genetics was still a young science. The concept of DNA had not yet been discovered, and much of what scientists knew about heredity came from studying how traits were passed down in plants and animals. McClintock, with her microscope and her maize plants, was about to change how people understood this mysterious process. She worked closely with Professor Rollins Emerson and another gifted student, George Beadle. Together, they became part of a small but passionate group of researchers exploring the inner workings of plant cells. Barbara's special focus was cytogenetics, the study of chromosomes, the thread-like structures inside cells that carry genetic information. While many researchers were content to record traits like the color of a kernel or the shape of a leaf, Barbara wanted to understand why those traits appeared and how chromosomes caused them. This required patience, skill, and a kind of scientific imagination that few possessed. She developed a technique for preparing slides of maize chromosomes that allowed her to observe them with remarkable clarity. Under her microscope, a new world opened. It was during these early years at Cornell that she made her first major discoveries. She found that chromosomes could physically break and rejoin, a process called recombination. This finding was more than just a curiosity. It showed that chromosomes were not fixed or static. They could change, they could repair, they could move, and in doing so, they could create variation, the fuel for evolution. Her work attracted attention. She was invited to present at conferences, and her papers were published in respected journals. But despite her achievements, McClintock was still a woman in a male-dominated field. She could not hold a formal faculty position at Cornell, not because she lacked talent, but because the university did not allow women on the faculty of the College of Agriculture at that time. It was a bitter truth, but Barbara didn't allow it to stop her. Instead of becoming angry or discouraged, she poured herself deeper into her research. She took on visiting research positions and fellowships. One such fellowship took her to the University of Missouri, where she would face even more institutional resistance, but also where she would make some of her most important discoveries. Back at Cornell, McClintock became known for her precision. In the field, she walked among rows of maize with a pencil and notebook in hand, carefully noting the position of each plant and recording tiny changes in color, shape, or growth. She often worked in silence, focused and alert. Her colleagues joked that she could hear the corn growing, so deep was her connection to the plants. She also trained her students and lab assistants to pay attention not just to data, but to the feel of the work, the way living systems sometimes resisted explanation and demanded intuition as well as logic. She believed that scientists should become part of the system they were studying, to watch and listen as much as they measured and counted. In those years, McClintock lived simply. She had no interest in fame, wealth, or comfort. Her small apartment was filled with books and dried maize specimens. She had few close friends, but many respected colleagues. For Barbara, the beauty of life's patterns was reward enough. One cold winter night, after hours of microscope work, Barbara stood at her window and looked out over the snow-covered campus. The world outside was still, but inside her mind, new ideas were growing, ideas that would later lead to the discovery of genetic elements that move within chromosomes. She didn't know it yet, but she was standing at the edge of a revolution in biology. Chapter 4. Mapping the Maize Genome. By the early 1930s, Barbara McClintock had become one of the leading cytogeneticists in the United States, even though few outside her field recognized her name. She wasn't chasing fame, she was chasing understanding. Every day, in greenhouses and laboratories, she worked with her maize plants and their mysterious inner workings. With every kernel she dissected and every slide she examined under her microscope, she was drawing closer to discoveries that would change genetics forever. At the heart of her work was one bold mission: to map the maize genome, to create a picture of how each trait connected to a specific part of the plant's chromosomes. This task was like solving a puzzle without a picture on the box. The only clues were the patterns of inheritance from one generation of plants to the next, and what she could see under the microscope. Barbara began by crossbreeding different types of corn and then observing their offspring. Some plants had purple kernels, others yellow. Some had smooth seeds, others were wrinkled. By carefully recording how traits appeared and disappeared over generations, Barbara could connect each trait to a specific position on a chromosome. It was incredibly detailed work, and one mistake could ruin an entire season of data. But McClintock thrived in this challenge. She used a process called chromosome mapping, a method still in its infancy at the time. This technique allowed her to place genes in a certain order on the chromosomes, based on how often they were inherited together. Genes that were close together on a chromosome tended to be passed down as a pair. Those farther apart were more likely to be separated during the process of recombination. Using this principle, Barbara slowly began building the first genetic maps of maize. But she didn't stop there. While studying abnormal patterns in her maize kernels, spotted, striped, and variegated, she realized something strange was happening. Certain traits did not behave the way traditional Mendelian genetics predicted. At first, she thought it was a mistake, but after careful rechecking and repeating her experiments, she became convinced something unusual was at work in the chromosomes themselves. Then came one of her most remarkable insights. She discovered that chromosomes could break and rejoin at specific locations. In particular, she identified a point on the chromosome that acted like an anchor, a place where the breakage occurred more often than anywhere else. She named it the centromere, while other scientists had described the centromere before. McClintock's work revealed how vital it was to understanding chromosome behavior during cell division. In one of her groundbreaking papers, she described how chromosomes could form unusual shapes, like rings or bridges, when they broke and rejoined incorrectly. These bridge breakage fusion cycles, as she called them, explained how genetic information could be rearranged within the cell, sometimes resulting in unexpected traits or even genetic instability. As she uncovered more secrets of the maize genome, her colleagues were astonished. Many had spent years trying to answer the same questions, but had gotten nowhere. Barbara, with her sharp eyes, deep focus, and love of complexity, seemed to see what others could not. Her work on mapping the maize genome reached its peak during a period when she received a fellowship to study at the California Institute of Technology. While there, she collaborated with other top scientists and further refined her techniques. She even explored other species' chromosomes, like fruit flies, to confirm that her findings weren't limited to maize. Despite her accomplishments, Barbara remained modest. She rarely spoke about her achievements unless someone asked directly. When presenting her findings at conferences, she spoke clearly but without flair. She wanted the science to speak for itself. Back in her Cornell greenhouse, surrounded by rows of carefully labeled plants, she continued her observations. The air was filled with the scent of damp soil and green leaves. Occasionally, she would pause, lean down to inspect a stalk, and smile to herself. She had learned to read the plants, to see stories written in the color of a kernel or the shape of a leaf. Each was a clue, a message from deep within the cell. As the decade closed, Barbara McClintock stood at the forefront of genetics. Her maize genome maps were among the most detailed and accurate of their time, but even more importantly, she had laid the foundation for what would become her most famous discovery: the existence of genetic elements that could move within and between chromosomes. The corn plants were whispering something new, and Barbara, as always, was listening. Chapter 5. Pioneering Discoveries. In Cytogenetics. By the early 1940s, Barbara McClintock had already gained quiet respect among a small circle of geneticists. Her discoveries in chromosome behavior and genome mapping had shown that she was not just skilled, she was exceptional. But Barbara wasn't done. Her mind remained restless, always asking questions no one else dared to ask. And it was during this time, while walking between the rows of her maize plants, that she noticed something that would eventually change the entire field of genetics. She had begun to observe odd patterns in corn kernels again, unexpected colors and markings that didn't match the predictions of classical genetics. Barbara knew her experiments were controlled, her observations precise. The plants were telling her something. She just had to figure out what it was. She went back to her microscope. Using her earlier techniques, she examined the chromosomes in cells from maize kernels that showed these unpredictable traits. There, she saw it. Tiny parts of chromosomes seemed to jump or move to new locations. At first, she doubted what she saw. Could genes move? No textbook had ever said so. Genes were supposed to stay fixed in place, lined up neatly on chromosomes like beads on a string. But Barbara saw something very different. In her maize plants, certain genetic elements were not fixed. They moved silently, unpredictably, creating changes in color and development. She called them controlling elements. She later named two of the most important ones, activator and dissociator. These were sequences of DNA that could switch other genes on or off depending on where they landed. When these elements jumped into or out of specific sites on the chromosome, they disrupted the normal activity of nearby genes. The result? Kernels with spots, streaks, or entirely unexpected colors. It was a revolutionary idea. What Barbara had discovered were what we now call transposable elements or jumping genes. She had found the first evidence that the genome was not a quiet, stable book of instructions, but a dynamic, interactive system. Still, few people believed her. When she presented her findings at scientific meetings, other researchers were puzzled. Her explanations didn't match their understanding of genetics, and many thought the idea of moving genes was too strange to be true. Some were polite, others skeptical. A few dismissed her work entirely, suggesting she must have made mistakes. Barbara, however, did not back down. She knew what she had seen, and she trusted her plans. Rather than trying to fight for recognition, she withdrew quietly from the spotlight and continued her experiments. For her, discovery was never about fame. It was about understanding how life worked from the inside out. She wrote detailed papers, carefully describing every step of her experiments and every pattern she saw. These documents, though difficult to understand for scientists unfamiliar with cytogenetics, were masterpieces of observation. Each slide, each kernel, and each field notebook entry was part of a story only Barbara seemed able to read. At Cold Spring Harbor Laboratory, where she had begun working full-time in 1941, she found an environment more suited to her quiet style. Located on the north shore of Long Island, New York, Cold Spring Harbor was a place of trees, sea breezes, and intense scientific focus. There, she had her own greenhouse and lab. She could work alone, without interruption. Most importantly, she could think freely. The staff respected her, even if they didn't always understand her. Many noticed her quiet strength and razor, sharp thinking. Some called her the Maize lady in admiration. She wasn't flashy, but when she spoke, people listened, if only because they sensed she was seeing something deeper than most could grasp. Outside of science, Barbara kept her life simple. She took long walks by the sea, tended to her plants, and sometimes played her banjo in private moments. She read widely philosophy, literature, poetry. Her curiosity wasn't limited to biology. She wanted to understand the world and her place within it. As the 1940s drew to a close, Barbara continued developing her theory of controlling elements. She published her work, explained it in lectures, and shared it with colleagues. But mainstream genetics still wasn't ready to accept such a bold idea. She could have become bitter. She could have stopped. But Barbara McClintock did what she had always done. She trusted her mind, her microscope, and most of all, the silent wisdom of her corn plants. The world was not yet ready to hear her, but one day it would be. Chapter 6: Silence and Solitude at Cold Spring Harbor. By the early 1950s, Barbara McClintock had retreated almost entirely from public scientific life, not because she had run out of ideas, far from it, but because the world of genetics had not yet caught up to hers. The concept of jumping genes, those mobile genetic elements that moved across chromosomes, remained too strange, too disruptive, too established theories. Few understood it, fewer accepted it. So Barbara stopped publishing. It was not an act of surrender. It was an act of preservation. She had grown weary of explaining her data again and again to people who did not want to believe it. Each seminar, each lecture, had become a battlefield. Instead, she chose something more powerful than argument: silence. At Cold Spring Harbor Laboratory, she found refuge. Nestled on the northern edge of Long Island, the institution was surrounded by trees, rocky beaches, and the quiet lapping of water. It was far from the noise of academia. And Barbara, always drawn to solitude, found comfort there. Cold Spring Harbor became more than her workplace. It became her sanctuary. Her days followed a rhythm few could match. Mornings began in the greenhouse, walking slowly between rows of maize plants. She would run her fingers along the leaves, check for subtle color shifts, and take notes in her worn brown notebook. No trait was too small to matter. A tiny spot on a kernel, a pale stripe on a stalk, these were clues to something deeper. In the afternoons, she returned to the lab. There, under the soft glow of her microscope lamp, she slipped into a world invisible to most. She prepared slides of maize root tips, skillfully staining and slicing them so that she could watch chromosomes in motion. The sight never bored her. It was like watching a drama unfold, a play directed by forces hidden inside every living cell. Visitors to the lab often found it quiet, almost sacred. Barbara did not welcome interruptions easily, but she wasn't unkind. She simply had no time for anything that took her away from her research. Occasionally, young scientists would ask to work with her. She chose only a few, and even then, she expected them to work not just with their hands, but with their minds. "You must hear what the material is saying to you," she once told a student. "You must let it tell you what to do, not the other way around." When she wasn't in the lab, Barbara often wandered along the shoreline, letting the sound of waves ease her thoughts. She loved nature deeply, not just as a scientist, but as a human being. Birds, trees, the sea, they gave her the same kind of quiet wisdom she found in chromosomes. Sometimes she would sit for hours on a bench near the bluff, watching the water shift with the wind. Though her professional world had grown small, her thinking had grown larger. The silence of rejection had not slowed her down. It had freed her. She no longer felt the need to explain her work to an unready audience. Instead, she continued exploring the behavior of controlling elements. She followed their patterns, their rhythms, their mysterious decisions to move or remain still. In her notebooks, she wrote reflections that sounded more like poetry than science. She spoke of genomes as dynamic systems, alive with motion and interaction. She began to understand that what she had seen in maize might be true across all life forms, that the genome was not a fixed blueprint, but a responsive, changing conversation between genes, environment, and time. By the mid-1950s, McClintock's work on transposable elements had reached astonishing depth, yet only a handful of scientists grasped its meaning. She was, in many ways, decades ahead of her time, and still, she worked. Each summer, new corn plants grew in the greenhouse. Each winter, new slides appeared under her microscope. Alone but never lonely, Barbara lived with a kind of quiet joy, a deep satisfaction in following truth, wherever it led her. In a world that often rewarded loud voices and quick results, she had chosen another path, one of silence, solitude, and slow, patient discovery. History had not yet caught up to Barbara McClintock, but it would. Chapter 7. Recognition Deferred. By the late 1950s and early 1960s, Barbara McClintock had become something of a mystery in the scientific community. Her name, once associated with bold discoveries and pioneering cytogenetics, now drifted quietly in the background. To many young researchers, she was simply the woman at Cold Spring Harbor who studied corn. They didn't know that in her notebooks, handwritten, deeply thoughtful, and filled with data-laden discoveries that would eventually reshape genetics. She still wasn't publishing much. She gave few talks. Some whispered that perhaps she had stopped working altogether, but they were wrong. Barbara was still at her microscope every day, still walking through her maize plots, still charting the movements of controlling elements with the same intensity as before. What had changed was the world around her. The field of genetics had moved on, racing toward the newly discovered double helix of DNA. Molecular biology was the future, and her quiet work with maize seemed to many like something from another era. But McClintock wasn't chasing trends. She never had. She was still interested in the bigger questions: how organisms adapt, how genes behave in real time, how life organizes itself. The tools may have changed, but her vision remained as sharp and ahead of its time as ever. Occasionally, someone would stumble upon her papers from the 1940s and early 50s and find them difficult to understand. She hadn't written for a general audience. Her language was precise, technical, and rich with observations from years of cytogenetic work. To truly understand Barbara's writing, you had to have seen what she saw under the microscope. You had to live among the maize, as she did. Some of the young scientists at Cold Spring Harbor, however, began to sense that something extraordinary had happened in her work. A few started to read her old papers carefully. They asked her questions. She responded patiently, though never in a boastful tone. She didn't claim importance for herself. If others understood her discoveries, that was good. If they didn't, she wasn't going to force it. But the truth, as always, has a way of surfacing. In the early 1960s, scientists working in bacteria and viruses began to observe behavior that mirrored what McClintock had described decades earlier in Maize. They saw genes switching on and off, moving from one location to another, just as Barbara's controlling elements had done. Slowly, the field began to realize that her findings were not strange exceptions, but fundamental truths. Even still, recognition came slowly. McClintock continued working, quietly, and rarely commented on the growing validation of her research. It was never about being right for her. It was about being thorough, about listening to what nature had to say. Then, in 1967, something remarkable happened. She was invited to deliver the prestigious presidential address for the Genetics Society of America. It was an honor that no one could ignore. When she stepped up to the podium, people were reminded of her brilliance. Her words, thoughtful and precise, drew from a lifetime of careful observation and unshakable dedication. More invitations followed. Awards began to trickle in. First, national honors like the National Medal of Science in 1970. Then, honorary degrees from respected universities. Articles appeared about her in scientific journals and popular magazines. People who had once ignored or misunderstood her began to listen. And yet, even as recognition finally arrived, Barbara remained unchanged. She continued her work at Cold Spring Harbor, still wearing her simple clothes, still walking slowly between her rows of maize. When asked how it felt to be celebrated after so many years, she smiled and simply said, "It's nice." She never let fame alter her rhythm, while the scientific community adjusted its view. Barbara stayed focused on the same mystery that had captivated her as a young woman: the dynamic nature of the genome, the intelligence of living systems, the dance of chromosomes under her microscope's lens. For decades, she had worked in solitude, sure of her findings, patient with the world's doubts. Now, at last, the world had begun to catch up. Chapter 8: The Nobel Moment. It was October 10, 1983, and Barbara McClintock, now 81 years old, sat alone in her quiet office at Cold Spring Harbor when the telephone rang. The voice on the other end was calm, slightly formal, and carried the distinct accent of Sweden. The words were simple, yet their meaning rang louder than any public applause she had ever received. "You have won the Nobel Prize in Physiology or Medicine." For a moment, Barbara didn't say anything. Then she laughed gently, the sound soft and surprised. After decades of quiet labor, decades during which her discoveries had been doubted, misunderstood, or dismissed, the world's most prestigious scientific honor had finally arrived. And it was hers alone. Barbara McClintock had become the first woman to receive an unshared Nobel Prize in Physiology or Medicine. The Nobel Committee cited her discovery of mobile genetic elements, what she had once called controlling elements in maize, long before DNA sequencing existed, long before her ideas were accepted. What she had seen under her microscope in the 1940s and 50s was now recognized as one of the most groundbreaking revelations in biology. And yet, Barbara took the news with the same quiet grace that had defined her entire career. When reporters arrived at Cold Spring Harbor, they found a scientist who had never sought the spotlight, who still preferred speaking with her plants over speaking to the press. Her workspace was modest, filled with notebooks, books, dried maize, stalks, and microscope slides. The woman they saw was sharp-eyed, composed, and radiating the strength of a lifetime's discipline. "I was astonished," she told them simply. "When asked how it felt to win, it never entered my mind." As the news spread, Tributes poured in from around the world. Scientific communities that had once overlooked her now praised her brilliance. Universities offered honorary degrees. Young geneticists, many of whom had grown up studying molecular biology, suddenly found themselves looking back at McClintock's early papers with fresh eyes and deep admiration. But Barbara was never interested in personal fame. Her joy came from the work itself. She was happiest among her maize, in the greenhouse and lab, where she had always felt most at home. Even after the Nobel Prize, she continued to walk those familiar rows, bend close to the plants, and study the quiet clues hidden in every leaf and kernel. The Nobel ceremony in Stockholm that December was elegant, formal, and filled with celebration. When Barbara stepped onto the grand stage, wearing a long dark gown with a pale shawl, she appeared both dignified and calm. She accepted the medal with a steady hand and offered her thanks with humility. In her Nobel lecture, she did not boast. Instead, she spoke about the genome not as a fixed instruction book, but as a system sensitive to stress and capable of change. She explained that genomes have ways of responding to the environment, of reorganizing themselves just as she had discovered through her maize plants decades earlier. To many in the audience, her words were startling. She described the genome almost as if it were alive, a system capable of communication within itself. It was a deeply advanced view, one that would take decades more to fully understand. But Barbara had been there all along, listening patiently while the world caught up. After returning from Stockholm, Barbara found her life a little busier. Invitations poured in. Lectures, interviews, celebrations. She accepted a few, always careful to protect her solitude. The attention was new, but it did not change her. She still rode her bicycle around Cold Spring Harbor, still played the banjo in quiet moments, still spent her days observing plants with the same intense wonder she'd felt as a child. She often said she didn't feel any different. "You can't put it into words," she said once about winning the Nobel. "It's like being lifted up into the clouds." For Barbara McClintock, the Nobel Prize was not a final destination. It was simply a recognition of what she had known all along, that patience, deep listening, and faith in nature could lead to truths that changed the world. She never stopped being the scientist who listened to her corn. Chapter 9. Legacy in Bloom. In the years following her Nobel Prize, Barbara McClintock became a symbol of brilliance, of perseverance, of the quiet power of original thought. But to those who knew her, she remained much the same as she had always been, a solitary observer, deeply in love with the living world, still following the whispers of Maize. Recognition didn't change her pace. Even in her 80s, she remained a regular presence at Cold Spring Harbor Laboratory. Her posture was a little more stooped, her hands a bit slower, but her mind sharp as ever. Visitors who came expecting a grand celebrity often left surprised. They had met someone who listened more than she spoke, someone who preferred ideas to attention. Barbara was generous with young scientists. She didn't hand out simple answers. Instead, she asked thoughtful questions that sent them back to their microscopes, wondering, exploring, thinking more deeply than before. She believed that discovery didn't come from rushing but from stillness. "You must have the time to look," she often said, "and the patience to hear what it is telling you." By the late 1980s, the scientific world had fully absorbed what she had seen long ago. The idea of a dynamic genome, one that could change, move, and adapt, had become central to modern genetics. Transposable elements, once viewed as strange anomalies, were now understood to play key roles in evolution, development, and even disease. Research into human DNA revealed mobile genetic elements that echoed Barbara's Maize discoveries. The entire field had bent in her direction, and yet she didn't gloat. She never pointed out how long it had taken. She had always trusted the truth to stand on its own. Her later years were spent mostly at Cold Spring Harbor. She lived in a small, modest house on the grounds, not far from her greenhouse. On warm days, she would walk along the gravel paths, greeting staff with a nod or a smile. In the evenings, she often sat by the water, watching the sunset over Long Island Sound. She continued reading widely, poetry, philosophy, history. She remained deeply curious about how living systems worked, not just in corn, but in all of life. Sometimes, when asked how she saw science and nature, she spoke almost poetically. A feeling for the organism, she would say. That phrase became associated with her, not just as a scientific idea, but as a way of seeing the world, with empathy, care, and respect for its hidden order. In 1986, the biography A Feeling for the Organism by Evelyn Fox Keller brought her story to a wider audience. The book described not just her discoveries, but her personality, her discipline, and her deep connection to her work. It introduced Barbara to readers outside the lab, women in science, students, artists, thinkers who saw in her a quiet hero. As interest in her life grew, so did admiration for the way she had lived it. She had refused to chase fame, had endured isolation and misunderstanding, and had remained devoted to truth for its own sake. She had shown that it was possible to live a life of purpose without loudness, a life of greatness without grandstanding. In the early 1990s, Barbara's health began to decline. She still visited her lab occasionally, though she needed more rest than before. Staff at Cold Spring Harbor cared for her with affection and respect. Everyone there knew they were in the presence of someone rare. On a clear summer day, when asked how she hoped to be remembered, she replied with a small shrug, as someone who looked and listened. Barbara McClintock passed away on September 2, 1992. At the age of 90, her death marked the end of a singular life, but her legacy had only just begun to bloom. Her work continues to inspire generations of scientists who study not just what genes are, but what they do, how they move, adapt, respond. In Maize, in humans, in every living organism, the echoes of her discoveries live on. And in quiet greenhouses around the world, where young researchers bend over microscopes and listen carefully to their specimens, Barbara's spirit is still there, not loudly, not obviously, but unmistakably present. Chapter 10. Seeds of the Future. Barbara McClintock left no children, no memoir, no grand public farewell. Yet what she left behind has taken root in the minds and work of thousands. Her life, quiet, observant, and defiantly unconventional, planted seeds that continue to grow in science, in education, and in how we understand nature itself. In the decades since her passing in 1992, Barbara's influence has only deepened. In laboratories across the world, researchers explored the dynamics of the genome in ways she had once only imagined through her Maize slides and notebooks. Transposable elements, which she first saw darting through corn chromosomes, are now recognized not as biological oddities but as powerful drivers of genetic diversity and evolution. Scientists have linked these jumping genes to the development of certain cancers, immune responses, and even neurological processes. They're no longer silent outliers. They are central to understanding how life changes, adapts, and sometimes protects itself from within. What Barbara discovered in corn has become vital to human health, biotechnology, and evolutionary theory. But perhaps her greatest legacy is not in the findings alone, but in the way she found them. Barbara's method, careful, patient, guided by intuition as much as by logic, has inspired a new generation of scientists to slow down, to look longer, to listen more closely. In a scientific era driven by rapid results and publication metrics, her life is a reminder of the power of deep attention. Institutions have honored her. Buildings at Cold Spring Harbor Laboratory now bear her name. So do awards, lectureships, and scholarships around the world. Her portrait hangs in scientific halls once closed to women. Students read her papers in graduate courses and marvel at how a woman, working mostly alone with Maize reshaped biology. More than a symbol of scientific brilliance, Barbara McClintock has become a beacon for those who find joy in curiosity, who ask questions no one else is asking, who are willing to wait for answers that others miss. For women in science, her story carries particular weight. She succeeded not by conforming to the expectations of her time, but by refusing them, by trusting her own mind and her own eyes. She showed that independence was not isolation, that solitude was not loneliness, and that scientific discovery could come from listening as much as from declaring. The phrase she once used to describe her approach, a feeling for the organism, has become something larger. It suggests a way of doing science that is empathetic, not mechanical, intuitive, not just analytical. It honors the living systems scientists study, acknowledging that they are not static puzzles, but responsive, evolving conversations. And in this, Barbara's legacy lives far beyond Maize, beyond genetics, beyond any single field. It lives in how we understand growth, of organisms, of ideas, of individuals. Every time a young scientist decides to stay with a difficult problem a little longer, to trust what they observe rather than what others expect, Barbara is present. Every time someone chooses to work quietly, without needing praise, she is there. Every time a woman in a lab believes she can lead a discovery, even if she walks alone, Barbara walks with her. In the end, Barbara McClintock's life wasn't just about discovering the motion of genes. It was about showing that truth. Deep, lasting truth often grows from solitude, from patience, from faith in what you see when no one else sees it yet. Like a seed buried deep in the soil, it takes time to rise. But it does, and when it does, it blooms. Thanks for learning with Ku English. Don't forget to like, share, and subscribe. Until next time.